In this section of Chapter 3, we're going to be dealing with carbohydrates. Carbohydrates are often called sugars. These carbohydrates kind of get their name because of the ratio of atoms in their composition. One carbon, two hydrogens, and one oxygen, CH2O, for carbohydrates. If we look at their basic functions, many of these carbohydrates are useful as energy sources in our body, and many of them are quick energy sources. And what this refers to is their ability to be easily catabolized to liberate energy that's going to be used to make ATPs. Another important function of these carbohydrates is that in many instances, some of the larger ones compose important structural building materials in cells. So, for example, cellulose in plant cells. Now, as polymers, carbohydrates are built from simple sugars, and we call these monosaccharides. So monosaccharides form the monomers of polysaccharides. And again, please review the dehydration synthesis hydrolysis reaction set to see how we build these and tear these down. So let's talk about some monosaccharides. Probably the most common monosaccharide is glucose. And then as a little side note here, remember that most carbohydrates have the ending OSE, so glucose, maltose, fructose, etc. These are all sugars. Now, glucose is the most common monosaccharide. It's often referenced in your blood sugar level because, again, it is very easy to catabolize and break down. And we'll spend an entire chapter talking about this process in a little bit later on in the semester. If we look at the glucose sugar, we see that its atomic composition is C6H12O6, and if we count these 6 carbons, 1, 2, 3, 4, 5, 6, we see they sort of form this ring, right? So this is a 6-carbon sugar. Again, the most common, probably, monosaccharide. Now, if we look at how we form disaccharides and ultimately polysaccharides by dehydration synthesis, the covalent bond that is formed when dehydration synthesis reactions link these monosaccharides together is called a glycosidic linkage. So here we can see an example of 2 monosaccharides being joined together by dehydration synthesis to form a disaccharide. So here's glucose and fructose monosaccharides, and notice we're pulling this water molecule off of here during dehydration synthesis. Removing this water molecule creates this special bond called a glycosidic linkage here that holds the glucose and fructose together, forming sucrose, which is a disaccharide. Some other common disaccharides are maltose that you find in beer, and lactose, that is the common sugar found in milk. Now, the large polysaccharides are macromolecules that consist of a few hundred to a few thousand monosaccharides joined together. They're useful for storage. By putting many of these monosaccharides together, they can be catabolized using the hydrolysis reaction to create monosaccharides that can in turn be used as a fuel source to make ATPs. We also see many of these large polysaccharides useful for building materials inside of cells. So here's a couple of examples of some storage polysaccharides. At the top is starch, and we can see that it consists of thousands and thousands and thousands of these simple monosaccharides joined together by these glycosidic linkages. Now, starch is the very, very common plant storage polysaccharide, while in animals, the storage polysaccharide is called glycogen. And again, very similar, simple glucose is kind of held together here by these glycosidic linkages. Glucogen is held in our muscles, stored in our muscle cells, for example, so that it can be used quickly when your body needs a quick energy source. An example of a structural set of polysaccharides would be cellulose that we see here in plant cells and chitin that we find in animal cells. So let's look at cellulose first. If we look at cellulose, we see that it's, again, long threads of these monosaccharides held together, forming these individual strands. But notice that in the form of cellulose, we see hydrogen bonding that occurs between these strands. Now recall that hydrogen bonds are bonds between a partially positive hydrogen atom and something that's partially negative on another molecule. These hydrogen bonds individually are not very strong, but when we put a whole bunch of them collectively together, it gives these molecules tremendous structural strength. And this is why we see these threads kind of being woven together here, forming the cellulose cell walls of plant cells that gives them their rigid shape. If we look at the example of chitin found in an animal's body, chitin is not found in all animal bodies, but it's especially common in the exoskeleton of what are called arthropods. Now, arthropods are, as we'll learn next semester, are animals with jointed appendages and an exoskeleton on the outside of their body. This exoskeleton made of chitin hardens after it is secreted, and it's the crunch that you feel when you crack into a crawfish or a lobster making their outer body shell. The chitin monomer has a sugar with a special amino group on it right here that gives it some additional structural strength, and this chitin is secreted as a liquid, and over time it hardens to form that hard exoskeleton.